Hey everyone, Manchak here again with another video for you in concepts C8 and C9. So for you Bio 30s, you should have already done the first video in this section on concept C8, which had us looking at how DNA is the hereditary material and how we got to that realization. What took us down that path to realize that. What we're going to do in this video, hopefully a relatively quick one, is to really quickly go through and talk about the structure of DNA. And thanks to Watson, Crick, Franklin, and all those other ones that helped out to discover that. And then we're going to move into talking about how DNA replicates and the process that takes place in there. So this stuff gets a little bit technical. You're definitely going to have to be on your ball as you try, or on the ball, to try to learn this stuff. Watch out for a lot of terminology in this section. So things like 5' and 3', lagging versus leading, DNA polymerase, DNA ligase, all of those kinds of things are going to come up. I would really strongly recommend to you that you have a look at the Amoeba Sisters DNA replication video. It is excellent. They do a really good job of working our way through the DNA replication process and spend some time at the beginning introducing you to the characters that are involved in this process. So I'm going to jump into the lesson so that we can keep this relatively straightforward. Please make sure you have your notes out and you're following along with me. So we are, by the way, transitioning into the third section of Unit C. I've kind of broken the section up into mitosis and meiosis. That's Section 1. Section 2 is classical genetics. And then Section 3 of the unit is molecular genetics, which is what we're getting into now. So we're really going to start to look at genetics and DNA at the molecular level and try to get a sense in concept C9 what DNA is made up of, and the process of DNA replication that sets a cell up to divide. What we're going to do in subsequent sections is we're going to have a look at what else DNA does for us, which is code for amino acids, which can be, or code for proteins, which can be made into the different structures that we find inside of our cells that give our cells structure and allow us to have a certain function, those kinds of things. So we're going to explore the basic structure of deoxyribonucleic acid, or DNA, and its role in protein synthesis. And then D11, our last C11, pardon me, our last concept in this unit is going to have a look at mutation and what goes on with DNA mutation and why that's something that's important for us to pay attention to. So I'm going to skip over the focusing questions because we did go over that in the last video, the first video in this week's worth of stuff, and I'm going to jump right into working through the structure of DNA. So a couple of things to remember before we get started. Number one, the nucleus of every cell in our body contains deoxyribonucleic acid, or DNA. And yeah, I want you to know both terms, okay? So be able to throw out deoxyribonucleic acid when you need to, be able to throw out DNA when you need to, okay? DNA is the only molecule that is capable of replicating itself. Sugars and fats cannot replicate themselves, but DNA can, and it's got a whole host of supporting structures that allow it to do that. But in order to manufacture a fat or a protein, we need a blueprint of some sort, so there has to be something else that's participating in that process. And DNA replication is essential for cell division. We know that in order for cells to end up with the right number of chromosomes, when we're done the process of either mitosis or meiosis, DNA replication had to have taken place to start with. So let's look at the DNA molecule itself. I'm going to describe these three things to you, and then we're going to get into actually looking at a physical structure of what DNA looks like. So the three parts that you need to know for a DNA molecule are the phosphate groups, the deoxyribose sugars, and the nitrogen bases. Okay, the phosphate groups, if you've taken any chemistry, you know phosphate to be PO4. The phosphate groups, along with the sugar, form what we call the backbone of the DNA molecule, and I'll talk to you about what that is in a second. The deoxyribose sugars, these molecules are very similar to the ones that are found in RNA, except that this one is lacking a hydroxyl group. So that's the deoxy. The hydroxyl is off of the deoxyribose sugar. In RNA, which is ribonucleic acid, notice it doesn't have the deoxy at the beginning of it. In RNA, they have that hydroxyl group attached to them, and that's why we call them a ribose sugar instead of a deoxyribose sugar. And then the nitrogen bases are really where the action takes place. Okay, the nitrogen bases are really the key. They are in those nitrogen bases is where our blueprint lies for whatever organism we're dealing with. Every organism has DNA inside of it, and that DNA is the blueprint for the proteins that make the organism do things and appear the way they're supposed to. So there's 4 nitrogen bases, and this is so incredibly mind-boggling when you really boil it down. This is so incredibly mind-boggling. Only 4 nitrogen bases in DNA across the world. In all organisms, and those 4 nitrogen bases and how they are arranged inside organisms makes everybody different. Okay, so these nitrogen bases are adenine, thymine, cytosine, and guanine, or A, T, C, and G. Again, I want you guys to know the name adenine, thymine, cytosine, guanine, and I want you to know that adenine is A, thymine is T, cytosine is C, guanine is G. Each one of these, if you combine a phosphate, a deoxyribose sugar, and a nitrogen base, you get what's called a nucleotide. Okay, and I'm going to show a diagram in a second here, and we'll kind of try to square that circle for you. Okay, so let's jump to the next slide. This is the structure of DNA. Now, I don't need you guys to know this in this complicated a way. I want something a little simpler from you. And I'll draw something in my horrible drawing with my mouse in a second here. But let's just look at what's going on in here. First off, I'll just use the highlighter for now. First off, this section over here on this side and this section over here on this side, this is our backbone of the DNA molecule, quote unquote backbone. And you'll notice that it is alternating phosphate, sugar, phosphate, sugar, phosphate, sugar. So this 5-sided structure in here, this is your deoxyribose sugar. Okay, and we've got an attachment point here. There's actually a carbon there. Those of you that are doing Chem 30 right now, you're going to see this. Those that you do Chem 30 later on, you'll kind of get a little more comfortable with what these diagrams are. So we've got our backbone here of alternating phosphate, sugar, phosphate, sugar, phosphate, sugar, and this one does the same thing except that it's phosphate, sugar, phosphate, sugar. It's going opposite. So I'm going to introduce this 5' and 3' thing right now. 5' and 3' are just a way for us to describe which end of the DNA molecule we're dealing with. The 5' end is the end that has the phosphate group or terminates in a phosphate group, and the 3' end is the end that terminates in a sugar, the deoxyribose sugar. So this is the 5' end and the 3' end of this side, okay? But what we have is something that is opposite on the other end. Okay, and this is going to become important when we look at replication and synthesis later on, is that if the 5' end is on this side of the ladder, then on this side is the 3' end. Okay, and then that means that the 5' end is down here over on this side. Attached to the sugars, okay, attached to the sugars are your nitrogen bases, and we've got our 4 representatives here. In this case, this is adenine in green, thymine in purple, guanine in blue, and cytosine in pinky red. And those nitrogen bases are attached to another nitrogen base. And so we have paired nitrogen bases that sit together. And what you guys are going to notice, and what is super, super important as we go on, is that adenine will always be paired with thymine. Let's try that again. Adenine will always be paired with thymine, and guanine or cytosine will always be paired with guanine. Okay? And these nitrogen bases are paired and held together by these dotted lines here. And those dotted lines, for those of you that have taken some chemistry, you might find this interesting. These dotted lines here are hydrogen bonds, okay? And so hydrogen bonds, we know, are not a true bond. It's more of an intermolecular force, which means that they can be broken relatively easily, but it is the strongest of the hydrogen or of the intermolecular forces. One set, and I've talked about this already, one set of phosphate, sugar, and nitrogen base is called a nucleotide. And so on this diagram, we have 1, 2, 3, 4, 5, 6, 7, 8 nucleotides. And they are paired complementarily, which means that if we have an adenine nucleotide over here, what complements it is a thymine. If we have a cytosine nucleotide over here, we have to have a guanine over here. Okay? So that's the general structure of the DNA molecule. If I ask you to draw a structure of a DNA molecule, first off, I will never ask you to draw in this much detail, as you see up here. I want to make it simple. You would start out and say, well, maybe a P represents a phosphate, right? I'm just going to go and just write out phosphate so you guys are sure what I'm talking about. Okay, and then maybe an S, and we'll put maybe a square around it. That's going to represent your sugar. And then you're going to have something that will represent your adenine, for example. That might be an adenine. And then a structure that might fit into that would be a thymine, and so on. Okay, so if I ask you guys, and I will at some point, to draw me a simple structure of a DNA molecule, I want to see something like this. You know, it doesn't have to be super advanced, but I'm going to be watching out. Usually I draw my sugars as pentagons because I know that this is a 5-sided sugar. Okay, so I'm going to have a phosphorus and a salt and a sugar attached to maybe an adenine here. Okay, my poorly drawn adenine. Okay, and then I'm going to continue on with my next phosphorus, which will attach to another sugar, 2, 3, 4, 5, which is going to maybe attach to a cytosine, which might look something like that. Okay, that is 2 nucleotides, but then we need our complementary strand. That's the other side of what's going on here. So over here we would know we would have a thymine attached. That thymine would be attached to the sugar, 2, 3, 4, 5, which would be attached to a phosphate over here, and so on up there. There's my sugar, another phosphate, right? And that's the level of drawing that I want from you guys. So when I ask you to do this, I want you to be able to make me a diagram that looks something like this, hopefully better drawn than what I've been able to do here. And the things that I'm going to be watching out for, okay? Alternating phosphorus and sugar, phosphate, sugar, phosphate, sugar. I'm going to be watching out to make sure you've paired your nitrogen bases properly. So adenine will be paired with thymine, cytosine will be paired with guanine. I'm going to check your complementary strand to make sure that's okay. And I'm going to make sure that you guys have a little bit of a legend up here that illustrates what each of the things represent. Okay, so a legend. I'm going to check the backbone. I'm going to make sure you've got complementary strands going on. And for an added bonus, if you're able to, you could put 5' and 3', you know, and then this one maybe we'll finish off with the phosphate here. And this will be, whoops, this will be 5', and this will be a 3', and I would just add a sugar over here. Okay, so I hope that makes sense to you. This is what I want. I don't need all of this complicated diagram that we have going on here. You can simplify it down, but I will ask you at some point to replicate the structure of DNA. So in a DNA molecule, the bases of one strand of DNA are paired with the bases of the other strand. Okay, adenine is always paired with thymine, guanine always with cytosine. We call this complementary base pairing. Okay, complementary meaning A complements T, C complements G. In complementary base pairing, the purines A and G pair with pyrimidines, which are T and C. I don't want you to get too hung up on purines and pyrimidines. They're more or less describing the general structure of the compounds rather than describing them themselves. So, you know, that we said adenine and guanine are purines. You'll notice that they are a little more complicated. They've got 2 rings, right? And then the pyrimidines only have 1 ring. Again, don't get too hung up on that right now. You'll probably need some university level chemistry to really make sense of that, okay? The two strands of DNA are considered to be anti-parallel. So they're parallel, but they're anti because they are opposite to each other, right? That means the 5' end in one pairs with the 3' end in another. So this is a simple representation. If this is our 5' end of this DNA strand, and this is the 3' end of the complementary strand. And so that means that we have complementary base pairing going on, and you'll notice that we've got that going on here. And if this strand stretches from 5' to 3', then this strand needs to stretch from 3' to 5'. So that's your anti-parallel strand arrangement, okay? Remember, the 5' end has a terminal phosphate group, and the 3' end has a terminal hydroxyl group. Just for me, just think of it as a sugar group, right? Just think there's your terminal hydroxyl group there, but that's attached to your sugar. Just kind of think of it that way. I think it'll be a little easier for you at this point. The direction of the strand is important because enzymes read genes in order to copy the DNA or to create proteins, and they read in a particular sequence, okay? If we don't know the sequence of one strand of DNA, or sorry, let me rephrase that. If we know the sequence of one strand of DNA, we will automatically know the sequence of the other strand, right? If I was to go back up here and we were not given this strand here, but we knew the sequence of bases that were appearing here, like adenine, thymine, guanine, cytosine, etc., ATCG, right? Whatever it is here, I would automatically know what the complementary base pairing would be, because A always pairs with T, T always with A, G with C, C with G, G with C, right? And on and on and on down the line. So even though we only know one strand, we can figure out the complementary strand very easily just by remembering that A pairs with T, C pairs with G. Okay? So what we're going to get into now is I want to get into talking to you guys about DNA replication and what replication is all about. This is where it gets tricky. So I don't know how you want to do this. I'll give you two options. You can hit pause now and go and watch the Amoeba Sisters video that I put just above where you found this video. Or you can watch this and then watch the Amoeba Sisters video. It's entirely up to you. But I would recommend you do both because I'm going to wave my arms around and draw really poorly on the screen. They've done some excellent animation that is a little more colorful, and I think as good an explanation, if not better than my explanation. Although I hope I'll do a good job for you. You can let me know if you want. So DNA replication, we've already talked about this. We know that this is the process by which the DNA makes an exact copy of itself. And for cell division to occur properly, the DNA must replicate. We must make an exact copy of each of the chromosomes inside of each of the cells that are about to divide. DNA replication is the same in prokaryotes and eukaryotes. And I'm not going to dig too deep into prokaryote versus eukaryote. Just remember that one has to do with having a nucleus, one not, and there's a few other subtle differences in there that I believe the Amoeba Sisters go through pretty well for you. The DNA replication is considered to be what we call semi-conservative. That's an important term and something I would like you guys to highlight right now if you haven't already. Semi-conservative replication is the process of replication by which each DNA molecule is composed of, the new DNA molecules are composed of what I would consider to be old DNA and new DNA, okay? Old DNA means that this is what this was our template strand, okay, and the enzymes read the template strand and constructed a new strand and sandwiched it onto there to create complementary base pairing. But that when you duplicate the DNA, you start out with one strand and you end up with two strands of double strands of DNA. And in the process, we take that one strand and we separate the complementary bases, and then we replicate new strands on the inside. So we can consider it to be one parent strand, okay, and one newly synthesized strand. That's what we're dealing with here in this case. So one parent and one newly synthesized. That's why it's semi-conservative. We conserve one strand, to be original or parent DNA, but there is a new strand. If it was fully conservative, then I'm not exactly sure how that would end up happening. So this is what we call semi-conservative replication. This type of replication involves separating the two parent strands and using them to make new strands. So I'm going to slide this over here. That's what this diagram over here is illustrating. So this is our coiled up double helix structure of our DNA. And what ends up happening is during the process of replication, remember those hydrogen bonds that were holding together the nitrogen bases? Well, there's a little enzyme that runs up the DNA strand, and it basically unzips it, is the best way that I would describe it. And so you can see it's been unzipped down here, and it's still zipped up over here. And they're going to break the hydrogen bonds in here, and then we're going to replicate. Okay, and then we're going to have in blue would be your parent strand or original DNA, and then the green that's being created here, that's your new strand of DNA. This is why it's semi-conservative. What you guys will notice on here, and we'll get to this in a second, is that one side seems to be replicating in small chunks, whereas the other side is replicating continuously. Okay, this is what we call the leading strand over here that replicates continuously, and this is what's called the lagging strand, because it has to go, and as more unzips, it's got to start to fill in the blank. This one can continue to replicate continuously because it's just following the direction of unzipping. We'll get to that in a second. In order to do this, we must break the hydrogen bonds. We've talked about that, and unzip that double helix that we've got going on. And each single DNA strands, so this DNA strand over here and this one over here, is acting as a template to create an exact copy then, or complementary copy of what's going on each side. So this is what we mean by semi-conservative replication, is you start out with a double helix. When we're done, you'll see that the dark red here is our parent strand on either side, and then the newly synthesized is in pink, which might be barely visible for you at this point. So that's what semi-conservative replication is. What I'm going to do now is let's talk about how we're going to try to break down how this process of DNA replication takes place. Okay. There's going to be a lot of players involved here. So one of the big things you need to do to learn this section is take the time to learn each of the players involved. So that means understand what each of the enzymes are involved. And the way I usually try to help you to learn this is maybe do cue cards and write down a cue card for each of the enzymes, and then try to use those cue cards to assemble the process of DNA replication. And so that might involve, having a cue card that says leading strand and lagging strand, and then tuck underneath the cue cards of the enzymes that might be involved with that, or maybe put them in order sequentially of how that might work. Up to you how you want to do that. So, how this is going to work. We already said that in order for us to do semi-conservative replication, we first have to unwrap and unzip that DNA molecule in order for this to take place. So we can't do that without some aid, because those hydrogen bonds are relatively hard to break. So we use an enzyme. We know an enzyme are used in our bodies to make a reaction of some sort, or to happen under body conditions, or to make a process happen more easily with less input of energy. Think of them as a catalyst, right? And so the DNA helicase, which is the first enzyme we're going to talk about, works to unzip the double helix. So I always think of helicase. Remember, I told you in Bio 20, if an enzyme or something ends with ASE, usually it's going to be an enzyme. So a helicase, think of the helix, think of unzipping the helix. So DNA helicase is all about unzipping the DNA double helix. What happens then is to prevent the reforming of the bonds, because that little enzyme is going to run up the DNA strand and unwrap and unzip it. But what's going to happen is those hydrogen bonds are going to want to come back together and reattach. And so what's going to happen instead is there's going to be some just proteins that'll come in and bind in certain spots to prevent that helix from coming back together. So our DNA helicase has now unzipped a small section of the DNA to allow replication to begin. And that spot where the unzipping has been completed is a replication fork. So you can imagine the DNA molecule runs down, and then now it branches off, and there's a fork in the road. One template strand over here, one template strand over here. Now this is where it starts to get a little bit trickier. What we need to remember in this diagram, and this is the diagram I'll sort of use to try to illustrate what's going on in this section for you, just for some consistency. This is the coiled up double helix. You guys can ignore topoisomerase, that you don't need to know about that. There's our DNA helicase molecule that's just running up the middle and branching things and breaking those hydrogen bonds. And so we've got now this replication fork here. Here's our original DNA. Now we've got the fork that's forming. These are the proteins that prevent it from zipping back up. And then what we have is strands that run, and I'm going to just make sure I wrap this correctly. Notice that we've got 5' over here, and this strand wraps up, goes down, and continues along over here. So this is the 5' end of the bottom strand. This is the 3' end of the bottom strand. Up here on the top strand, it's 3' back to 5'. Okay. Now, one template runs 3' to 5'. Okay, and we're going to look at this is the 3' to 5' template on the bottom, our leading strand, and our lagging strand is the 5' to 3' strand. Okay, and there's a lot going on in here. When we're done, hopefully it'll make a bit of sense to you. So using the template strands, new strands are built using complementary pairing. New strands are synthesized using an enzyme called DNA polymerase 3. Okay, there is a DNA polymerase 1 as well, which we'll talk about, so be cautious. DNA polymerase 3 builds the new strand by linking bases that are complementary to what is there. So just imagine, I'm gonna sort of scoot back up to the last slide here. Here's our DNA polymerase 3. All it's doing is it's running along and it's pulling in nucleotides, and it's trying to find a match for whatever's coming up next. So if this long one here, let's say, is adenine, it's going to have to find a thymine to pop in on this spot. And if this short one here is a guanine, then it's going to have to find a cytosine to pair in here. And it's just going to run along in a 3' to 5' on the template strand direction, popping in little DNA nucleotides to do the complementary base pairing. The DNA polymerase up here is doing the same kind of thing, and it's moving in the same direction, which means that it's moving in the 3' to 5' on the template strand direction. Okay, so it's running that way. Sorry, that probably didn't make sense. It's running this way on the strand from right to left here, but on this one, it's running from left to right. This is where we get into talking about leading and lagging strand. Okay. For DNA polymerase to start, there must be a primer attached to the template. A primer is a short piece of RNA that says, hey, start replicating here. Okay. The DNA polymerase starts at the primer, adding nucleotides in a 5' to 3' direction for the newly synthesized DNA. But it's reading the strand in a 3' to 5' direction. So it's constructing in 5' to 3', but reading in 3' to 5'. Maybe rewind and listen to that a couple of times so that it makes sense to you. What's great about the leading strand is because it reads in 3' to 5', the DNA helicase is unwinding in a 3' to 5' direction. So on the leading strand, the DNA polymerase can replicate continuously. It's just going to follow the helicase. Okay, because it reads in a 3' to 5' and builds 5' to 3'. The DNA polymerase does the same thing on the lagging strand, except that it's reading and moving in the opposite direction. So in the leading strand, it's going in one direction following the helicase. In the lagging strand, it's going in the opposite direction from the helicase. And so that makes a difference in what's going on between leading and lagging. So in the lagging strand, it's still synthesizing, but it's synthesizing in short fragments because it's still synthesizing 5' to 3', but it's reading 3' to 5'. And the DNA helicase is unwinding in the opposite direction now of which way the DNA polymerase needs to go on the lagging strand. Okay, I know it's complicated. I've got a couple of videos, and I think that the Amoeba Sisters video will help you out a fair bit too. Here's the process, just to run you through basically what's going on. And then we'll jump back to leading and lagging strand with the diagram in a second. So we need to add RNA primers as the, well, first thing that needs to happen is we need to unzip using the DNA helicase. Okay, then RNA primers are added. Those are marker points saying, hey, we need to start replicating here. Okay, then the DNA polymerase synthesizes a new strand from 5' to 3' on the new strand by reading the template strand 3' to 5'. The DNA primer is replaced with the DNA by something called DNA polymerase 1. So that little primer sits there as a marker, but eventually we're going to come to that point and go, well, we need DNA here, not RNA, because they're different chemicals. And so DNA polymerase 1 comes in and replaces that RNA and puts in the DNA nucleotides that need to be there. And then the last thing that needs to happen is DNA ligase, another enzyme, comes in and binds the gap between the fragments created by the RNA. In other words, it kind of ligase, it sticks things together. And we'll find another ligase later on in concept D5, I believe, or sorry, D5, in concept C10, I believe. And so DNA ligase is the glue. It sticks everything back together, zips everything, or not zips everything, sticks everything together to make sure all the bonds are where they need to be. And then, so this is the diagram that I'm going to sort of try to show you again here and try to make sense. So remember, the DNA polymerase operates by reading the template strand in a 3' to 5' direction. So on the bottom of our leading strand here, we have I'm going to try to get my pen up and running here. What kind of color do we want today? Let's go with a green. Over here then, looking at my parent strand is this one here. So there's my 3'. And over here, and it's not on the diagram, but there's my 5'. So what's happening is the DNA is reading the parent strand 3' to 5', but constructing the new strand, and here's your new strand right here, okay, constructing that new strand in a 5' to 3' direction, okay? And because it's reading 3' to 5', this parent strand here, as it unzips, as the DNA helicase unzips, this DNA polymerase can just follow along. Okay. However, what happens over here on the lagging strand is that we have to replicate in chunks because it's unzipping for this parent strand. Remember that this then is the, this then is the 3' end, and here is the 5' end of the lagging strand, okay? And so the DNA polymerase 3 still has to replicate by reading 3' to 5'. So it has to read in this direction, whereas the DNA polymerase here was reading in this direction. So I'm going to try to move to a different color that stands out a little bit differently. So we're reading in this direction on the lagging strand. And we're replicating then in a 5' to 3' way for the new strand. So what's happening, what needs to happen then, is as a little more unzips, we plop a couple of RNA primers in here, then the DNA polymerase attaches, and it runs its course until it bumps up against another RNA primer, then detaches, reattaches, runs its course, and basically what it's doing on the lagging strand is replicating in little short chunks, okay? That's why it lags. is it's not keeping up. It's not continuously replicating. It has to wait for some unzipping, pop a couple of RNA primers on, attach the DNA polymerase, let it run a bit until it bumps up into another RNA primer, detach, reattach, then circle back, find another RNA primer, do a little bit, right? And then detach and then go back. And we're going through a process of just doing little bits along the way. What this leaves us with then here is we've got these RNA chunks here that need to be replaced. And so what we end up with is, you'll remember we talked about the DNA polymerase 1 that swaps out those RNA pieces for DNA. And then our little DNA ligase right here comes along and it fills in any of the broken bonds here and seals those bonds back up. Okay, that was a lot of explanation for you. 35 minutes of me talking at you. DNA replication is tough. Probably the trickiest part of this section of the unit. So I would encourage you, you may need to go and watch that section again. I may have gone too quickly. I'm going to just quickly tab back to this so that I can remind you guys of a couple of things, okay? First off, okay, go and watch the DNA mechanism of DNA replication by the Amoeba sisters right here. Go and watch it. It's awesome. It'll help to explain things. This one right here, this mechanism of DNA replication advanced, it's 2 minutes and 18 seconds long. It's not a big commitment, but it's going to allow you to have a good idea of what this might actually look like in real time. I recommend watching it a couple of times and watch out for all the pieces. So maybe just pay attention to the leading strand to start with, and then run it through again and pay attention to the lagging strand. And watch for that big Okazaki fragment that sort of flies out, and then the DNA polymerase on the lagging strand comes and meets back up, and then it disconnects, and then another one flies out, and then it meets. It's really, really cool. And what blows my mind is that before a cell divides, this happens each and every time without us thinking about it, without any control. It just does it, and it's ridiculous, and it's amazing. Okay, so that is concept C9. DNA replication is tough. So please make sure you go back and spend a little extra time with it. If you're having trouble wrapping your head around it, let me know, and I'll try to do sort of a live explanation for you if I can and make it a little bit better. Watch some videos on it too. There's probably way better ways to explain it than I'm doing right now, but do be cautious. A lot of those videos might go into university level stuff that you guys aren't going to be held to. But if you can understand the university level stuff, then you'll be no problem understanding what we have to do in this class. After this, there are some questions in your textbook, and then a couple of practice worksheets I'm going to give you guys just to do some labeling and other bits and pieces associated with this. Then move on to your formative quiz, wait for my feedback, do your summative quiz. So that's all I have to say for concept C9. I hope you found this helpful. Stay healthy. Track me down if you have any questions, and we'll talk soon in concept C10.